but I'm one of the oldest. Dogs. Collagen. My name is Jeff Davidson. I had been at Vanderbilt for 35 years in the Department of Pathology, Immunology, and Microbiology. Bicycle riding.
I want to ride my bicycle. I want to ride my bike.
Collagen. There's a lot of it, and you can't live without it. Collagen obviously evolved as this triple helical molecule. And then my feeling is that there were a series of genetic variations over millions of years in much lower organisms that permitted the ability of larger animals, not only mammals but other animals, to survive and have resilient tissues, resilient protective tissues, also as a means for transmitting mechanical forces. I think the favorite discovery was actually not particularly on collagen, but on modifications of peptides, collagen peptides that could be used in micro nanoparticles to deliver other agents to wounds. Quite a bit, but I would say very often it was at the latter part of my work was wound repair in vivo, but with systems that were being used to evaluate the effects of various growth factors or gene sequences or other kinds of vectors to get variable outcomes in terms of wound healing. Well, it was always science. It was probably science from the time I was in second grade. But what brought me to these particular areas went through a variety of stages. I had initially in my PhD studied the development of sea urchin embryos. And there is some extracellular matrix there. But as time went on, and we moved into the laboratory of Paul Bornstein, who was a leader in collagen biology or matrix biology at the time, I began to identify new collagen types and their application or their application in different parts of biology of skin and other tissues. So there's a big spectrum of collagen molecules, a diverse spectrum. Paul Bornstein was my principal mentor. Subsequently at the NIH, I was in the laboratory of Ron Crystal, who was actually a lung biologist, but he was very interested in the aspects of collagen in lung development and lung pathology. I would say it was Bornstein. There was a major transition from what I was doing in marine biology and going into the Bornstein laboratory and looking at connective tissue biology. That's hard to define, but I think that there have been some actual papers actually developed here at Vanderbilt, which involved collaborations with people in biomedical engineering, and in which we were devising particles which could, microparticles which could be introduced into a wound site and can have sustained release of important growth factors or other agents. It's a good question. It's the diversification of matrix has grown enormously over the last 3 decades. So there's lots more details. Collagen is certainly not the only molecule, but it's the one that is quite stable. And there are different arrangements of collagen fibers in different tissues, different lifetimes, different sensitivities to proteolysis. So, and in particular, you can think about the differences in collagens between the basal lamina, which is a very delicate connective tissue, and the interstitial collagen fibers. I would say he did a pretty good job because collagen is, for at least for mammals or animals, is an essential factor. It's what connects us all together. And with severe defects in collagen, nothing happens. Find a good advisor. Oh, absolutely. It's not solved at all. There's a variety of features of that. One is, can we begin to modify collagen structure or collagen metabolism through molecular methods or chemical methods, but probably molecular. And the other is to consider more in terms of collagen fibers, to think about all the other associated molecules that have to be in that system in order to have an effective matrix. So the collagen is that stable cable, but it needs a lot of help. Well, it would have to go back to the early meetings when actually the collagen phenotypes were being identified suddenly at a rather rapid basis. And so it became clear that you can't simply talk about collagen. You have to issue a number tag at the end of that word to have the difference. And because they do have reasonably different mechanical properties, lifetimes, and so forth. So I got a lot of pleasure out of organizing meetings, and still do.